Hey, my name is Mitchell. I guess, okay, Professor Mike, I want to start off with saying this. I am going to basically be teaching this the same way you taught me in the review on Thursday before you let us out for reading week. Only creativity is I kind of went for a mad scientist theme with the lab coat and goggles. I don't know. Anyways, okay, so this is protein synthesis. Are you guys familiar with DNA? Like
Yeah.
A binds to T, C binds to G.
No.
No, I have no idea.
Okay, so quick introduction. DNA, you have two strands that are intertwined together into a double helix. You have A, T, C, and G. A binds to T and C binds to G. That's pretty much all you need to know from DNA for this. So, protein synthesis happens in the cell, more specifically in the nucleus of the cell. These are your two strands of DNA within the nucleus, and within these cells you have little letters, and so on and so forth. So this would be a T because that's an A, this would be an A because of T, G, and this would be a C. That's really bad. Basically, chicken scratch because it's such a small area. Anyways, so protein synthesis happens in two stages, transcription and translation. Transcription is when we add an extra piece of string, we don't know, it's called RNA, and it basically just copies all the letters off of here. The RNA then leaves the cell and goes to the ribosome of the cell. What it does is it goes through the cell like a train through a tunnel, and along these are little letters. So let's go C, C, A, U, G, A, C, U. Okay. So it's going to go through the ribosome, and in sets of 3, it's going to look for the start code, which is AUG. So it goes, is this AUG? No, that's CCA. Is this AUG? CAU. Nope. Is this AUG? Yes, AUG. So then it starts copying for the amino acid chain. And there are these things called T ribosomes that kind of look like clovers that are hanging around in the cell. And they have, okay, I guess I should have started with this. These letters here are called codons, and these here are anticodons. So to match with the codes of 3 along the RNA there. So the tRNA comes into the ribosome, matches up, and each T ribosome carries a different amino acid. So it comes into here, drops off the amino acid, and it starts making a chain. And then it just goes and picks up another amino acid for the same thing. The first one is always called methionine. Then the next T ribosome comes along for ACU, and it drops off, let's say that, and then that, creating that protein, strand of amino acid, and goes on, so on and so forth, until it reaches the kill code of 3 codons, which is UUA, UAG, and UGA. And then once it reaches that, it stops making proteins, and the chain is complete, and it goes off to your body to do that specific job. So, now, okay, stop it. All right, this is an example of your DNA strands. So 2 strands of DNA intertwined. So you have your code, which I already made to save time. Otherwise, I would have taken half an hour. Like that took me half an hour to do all that. So you have your code, which is start A, then there's like a G, and so on and so forth. And so what happens is the RNA comes in, copies itself to this one side of the DNA, and then leaves the cell. So these are now useless. So then this is your train of RNA that goes through the ribosome and starts looking for the start code. So is this AUG? No, no. This is AUG. So we'll start that with, let's say, a nib. So then that's a different amino acid. The T ribosome comes in carrying methionine, drops that off for this set, and so it's sets of 3 from now on. So then you have these 3, which let's say is that, then gummies, which is those three, and then so on and so forth all the way to the end. I'm sorry, Mike, I'm just going to be doing this for purposes. I'm not actually looking at the codes. So then this is this three. Let's do another three like that. And another three like that, and so on and so forth. And then that is your amino acid chain that goes to your body to do something. Because you can make different proteins for different things, like cooling off or warming up, stuff like that. So, one last thing to teach you is there are mutations within the DNA, or DNA and RNA. So there are two mutations, a point and a frame shift mutation. The point mutation, don't tell on me, is when one amino acid is different. So instead of this being white, which is U, because in RNA there is no T, it binds to U instead of T, let's say it's pink. And so now you have a completely different chain. So instead of being blue shark, jube is jube. And that's the one. And then there's also another one, which is frame shift mutation, which is the addition of another codon. So let's just say right there, let's just say it's a U. So then that now changes everything. So those for those three, that's for those three, that's for those three, that's for those three. Then you get another. Then everything is shifted. Like it's not the same three. The three codon chain is now different from before. I know that's a lot of information to cover in such a short time. I have no idea. Okay, let's just say within like 7 minutes. Do you guys have any questions?
No.
No, I just have to say you're like the best teacher.
Okay, you do not get points for sucking up. Like that does not factor into my grade.
I'm taking your marshmallows.
Okay, take them. They're for people eating. And cut.